Our protocol is significant because it allows for both the cost-effective and timely dereplication of known antibiotics without the use of specialized equipment. The main advantage to this technique is template customization. This means that an individual can tailor which resistance genes that they wish to include in their library template based on their desired level of broad or narrow substrate specificity. For example, a beta-lactamase expressing E. coli template can be prepared in order to allow for the highly specific dereplication of beta-lactams and their various subclasses.
Using aseptic technique, pipette 500 microliters of cation-adjusted MHB from a sterile reservoir into each well of a sterile 96 deep well plate. With the prepared ARP or MARP strain plates, use an applicator stick to inoculate the 96 deep well plate in accordance with the ARP or MARP map. Place a breathable sealing membrane on the surface of the deep well plate and incubate it overnight at 37 degrees Celsius, 250 rpm. Ensure that there are no contaminated wells. Using a multi-channel pipette, transfer 100 microliters from each well of the deep well plate to a sterile 96 well round bottom plate. Add 100 microliters of sterile 50% glycerol into each well and mix by gently pipetting up and down. Prepare at least 5 library plates. Cover the plates with sterile aluminum seals and ensure that each well is individually sealed. Place the plate lid on top of the aluminum seal and store at -80 degrees Celsius. Using a wooden applicator stick, gently